Thank you, RITAMBHARA. And also, I remember in that hackathon, I offered $10 per person if they join my team. So, so I don't know if that was the reason. I got very competitive at the time. So, but well, thank you so much for having me today. Really honored to be here. And this is actually my very first in-person seminar. So please be kind, because I forgot how to act in social situations. And as RITAMBHARA mentioned today, I'm going to talk about mainly privacy and to general overview, and then we can have a nice discussion, hopefully, at the end. So let's begin. Probably most of you know that what we are trying to do with genomics is kind of relate the genomes of patients or individuals to some kind of phenotypes. That could be a disease, or that could be any other characteristics. But now, to be able to do that, we have the technology to collect massive amount of data from other stuffs, such as epigenomes, transcriptomes, like gene expression or chromatin, and proteome and metabolome. And you can imagine if you're collecting this much information from individuals, this comes with a lot of challenges. Analysis is one of the challenges, such as how do we develop bioinformatics tools. But then there are these other challenges that people don't think as much, such as how do we share this data? What is the privacy implications? And how do we even move this large amount of data? So my research, one of the overarching goals of my research to answer the question, how do we understand the privacy problems in this collected data? It could be genomics data or just genetic data, or it could be epigenomics, transcriptomics, which we call functional genomics. How do we understand the privacy leakages, and then how do we kind of prevent these leakages? This is a bunch of studies that are either publicly funded or privately funded studies, or companies. They collect a massive amount of data from living individuals in some cases, and sometimes deceased individuals. And this data is every day being collected, and I don't know where we are at, but it's like a humongous amount of genomes and transcriptomes and epigenomes. And one of the biggest problems is the privacy, as I mentioned. And now why this is a problem is because genomes are our fingerprints, right? Even though we share 99% of our genomes with each other, there's this 1% difference that makes us different from everyone. And I always give this analogy of social security number because, I mean, it's not technically true, but social security numbers are unique to everyone, and it can open the doors to many other information about you. So genomes itself, yes, it tells something about your phenotypes, but it can also open the doors to understand some other stuff. But in general, what we learn from genomes are, this is like a typical what you get from 23andMe. You can tell if which geography you belong to. You can tell what kind of health predispositions you might have, your traits, things like wellness. But in addition to that, you can also use the genomes as keys to other databases to learn more medical information about individuals. And it was shown over and over that genomes cannot be de-identified because they are very unique. There are studies about, how you can find out your relatives or people's last names even. And also, DNA data breach is a little bit worse than the credit card, because you can change your credit card number, you cannot change your genome. So on top of the iceberg is what we usually see in the news, right? 23andMe is selling your data. You may not be able to get life insurance because of your DNA, and also Golden State Killer, where everyone, I think, by now heard about it. But actually, there is more to that. There are things related to genetic profiling, inclusion, and there are communities such as indigenous people that are being exploited. So genome privacy spans not only, the individual's monetary and dignitary harm, but also it relates to the trust between the patients and the institutions. And if you provide protection to privacy, then you can get more participation to genetic studies, and that will increase our statistical power to make meaningful discoveries. So that's why it's important. And also, genomes are not the only information that are private in the field of healthcare and medicine. And since we are at the Data Science Institute, I'm going to give an example from machine learning. So when we do training in machine learning in medicine, we have a bunch of patients. They have some observations. Observations are private data. And the outcomes of these observations are also private data. And moreover, the trained models can be private data because, especially if you use things like deep learning, they capture a lot of information in the training data that you can go back and figure out who is in the training data. And then when you do the inference, it's the same thing. You have a model, private or not. The input to this model is observations from a patient, which is private. And then the output is also some kind of prediction about the disease or anything else. It's private. So you can see that in every aspect of the data science, you run into these privacy problems that are a little bit overlooked, I guess. And now that we are moving the AI to medicine, like there are clinical decision support tools that uses machine learning, or we are making a lot of predictions, taking people's genomes and predicting stuff. You can imagine that there are privacy issues at every step of this pipeline, basically. So today, I want to tell you about my approach to this problem. So I have two-pronged approach in my lab. We first quantify what kind of private information there is in a given data type. And then we use this as a motivation to create algorithms to protect the private information during the sharing and the analysis of this data. And I'll start with the quantification first. So I'm assuming there are a bunch of computer scientists here. You probably heard of Netflix prize, or some of you perhaps too young to know what Netflix prize is. So this was a challenge that was put out by Netflix, where they gave anonymized movie preferences of their users, and you can do some kind of data science to figure out what we should suggest to them as new movies, right? Very, innocent way of looking at things. But then it was canceled because there was a privacy issue. And this privacy issue was brought to you by Arvind Narayanan, who is now at Princeton, but at the time was at UT Austin, I believe, where they took the Netflix data set, overlapped that data set in a statistically rigorous way with the IMDb user preferences. IMDb user preferences are not anonymized because it usually comes with the name of the individuals. And then you can actually de-anonymize the Netflix data doing that. And now once they show that, they cancel the price for the privacy issues. And when they were talking about this in the paper, there was two things that we were very interested in. That they talk about that these properties of this anonymized data set was sparse and noisy, and that's why their heuristic was working on it. And when we look at it, this sounds like a genomic study where it's very noisy and very sparse. And perhaps we can use the same way to do some kind of linking attack to understand what kind of private information leakage there is in different kinds of genomic signals, basically. So how does the linking attack work? In the very, very simple sense, you have two data sets. One is anonymized, one is not anonymized. And then you have common features in these two data sets. You overlap them. And then you basically figure out the private information about an individual in one that is in one data set and not in another data set. So this is a very simple way. This works when you have perfect overlap. But in genomics data and with other data sets, you may not have perfect overlap because the data is noisy and sparse. But before telling you how exactly statistically it works, I want to mention about what you can learn actually from the genetic data. Why do we care about these linking attacks, right? So for example, if you have someone's full genetic data, and if there's somewhere, let's say I somehow stole RITAMBHARA's genome, and then there's this data set somewhere, let's assume that it's people's genomes and their yearly income. I mean, that may not work, but let's, for the sake of example, I can overlap RITAMBHARA's genome in that database and then figure out how much she makes. If you have partial genomic data, then you can use the correlations that exist in the genome due to linkage disequilibrium and predict more information about the genome and then do the same attack. If you have summary level data, then it becomes a little bit harder to do this, but you can still figure out, let's say you're trying to figure out if a person participated in a drug abuse study, that is a GWAS study. If you have this person's genome, and if you know the allele frequencies of the SNPs that are correlating with the drug abuse, and then you know the population, the allele frequencies in the population, you can do some kind of comparative analysis, statistical analysis to figure out if that person participated in that study or not. So these are all examples of linking attacks. And then we have shown over the years during my postdoc and with other people in previously who was in the same lab at Gürsoy lab with me, we have shown that functional genomics data has a lot of problems with privacy that is not known because this data is usually collected to understand other biological signal, right? How the chromatin is open or closed, or are the genes are expressed or not. So that has nothing to do with the genetics. I mean, but we know that there's correlations to the genetics, and so you can use these correlations to figure out what kind of private information you have. And then I'm going to show you one example that I've done with RNA-seq data. So when you have RNA-seq reads, all of these reads comes with bits and pieces of the person's genome. The problem is that it's only 1% of the genome, and it's very noisy. So you get this noisy and sparse data set. And then if you steal someone's genome, and usually RNA-seq data is taken from a cohort, and this cohort might be a disease cohort. In this case, we are giving bipolar disorder as an example. If you steal that person's genome, you might be able to figure out which RNA-seq data belongs to this person, and then what is the phenotype of this person. And how this works is actually simple. We have a attack database D, which is very noisy, which is an RNA-seq data from a cohort of, let's say, 500 people. And now we have a stolen genome. We do genotyping and get this noisy genotype panel. It's very noisy. You can see precision and sensitivity is pretty low. And then we also have full genome from this individual's stolen genome. So we overlap every single individual in this panel with the full genome, and then score this intersection. We call it linking score, and this linking score is weighted by the allele frequency of the genotype. So if this person has a rare genotype, and it's in the panel, then you give higher score to the rare genotype because it gives you more information. And then we rank these individuals, and the assumption is that the top individual in that ranking is the individual that the target individual, right? And then we define a metric called gap that tells you the difference between the top individual and the rest of the cohort. And then you compare this gap with the randomly generated gap values, and if it's statistically significant, then you can confidently say that Jane is the 10th individual, and therefore her bipolar disorder phenotype is positive. So we did this for 400 individuals in GWADIS consortium that has overlapped with 1,000 Genomes project, and we found everyone, 100% accuracy. And then we asked the question, what if we downsample the RNA-seq data and keep doing that? And also we said, okay, how about I add some false positive genotypes to the data, so kind of noise the data even more. And as you can see, we can add up to 1 million false positive genotypes and go down to 5% subsampling. And you can see that for most of the time, right before 1 million false positives, for 10-25% subsampling, you still have 100% accuracy. So it's very robust because rare genotypes, if they are rare, it's very identifying. So this was sort of, yes?
Sorry, I'm not sure if I understand the noisy part correctly. Is that noise in terms of like the percentage of SNPs that are correctly inferred from the RNA-seq?
Yeah. So because RNA-seq has a lot of other artifacts like RNA editing, and then there is the sequencing artifacts. So RNA, you do usually sequencing on the cDNA, and then that conversion sometimes has problems. So you get a lot of PCR bias, all sorts of sequencing artifacts. It's more noisy than a regular whole genome sequencing data. And then we further added more noise to it. So it's very robust. And now we ask the question, okay, yeah, great, you did this, but you stole someone's genome. That's not possible with the current protections, right? So what I did is I went around with consent, grabbed coffee cups from individuals, and then I also withdraw their blood. So I did RNA-seq to their blood and add them to this anonymized database. And then I sequenced their coffee cup, not in as rigorous as law enforcement does it, but, you know, just bought a kit, went to the wet lab, you know, wipe off the coffee cup, and then got some genomes. It's very noisy too, but then this was because in US, you can actually grab discarded items and sequence it, so it's legal, completely legal. So I didn't do anything illegal, and also they were consented. But when we did that, and we also did subsampling, imputation, all sorts of different stuff to, you know, noise the data or enhance the signal, no matter what we did, most of the cases you can link someone's coffee cup to their RNA-seq data. So then we realized that regardless of the noise, the genetic signal is too high to ignore that this data can be shared publicly, basically. So this was the story about how we do linking attacks to quantify the private information leakage. But you can also imagine this is a lot of work, you know. So then we thought that perhaps we can use statistics and not do this whole linking attack study, but figure out a way to predict how much privacy leakage there is. So I developed this Gaussian regression model, Gaussian process regression model, where you take statistical properties of the RNA-seq or attack-seq data, basically the coverage, depth of sequencing, and how skewed the sequencing is, and then use it as an input, and then it tells you how many potential rare variants you will get from that data. And it's pretty accurate, actually. Here I'm testing on scikit encode data. Yes, here. And then in this study, we used a little bit of a population genetics approach where we used the hidden Markov models, the famous hidden Markov models to impute the genomes. But here, what we are trying to do is, if you have 10 to 20 common SNPs, what can you learn about this individual? And we were able to do direct identification, kinship analysis, even PRS scores for these individuals. And this was mostly done by my friend Prashant. So then we figure out these a little bit faster ways of quantifying information. But then this was kind of an inspiration to me to, you know, perhaps machine learning can help to understand how much privacy leakage in a data set. And whenever I show this RNA-seq study, everyone comes back to me and says, how about single-cell RNA-seq? Because now it's most common. So if you have single-cell RNA-seq count matrices, can you get any type of genotypes? So this is the recent study that I launched in my lab with my postdoc and with RITAMBHARA, where we are trying to take the cell by gene matrices, combine it with some other features to predict genotypes for the individuals. And so far, we have 80% accuracy, so we can actually accurately predict. But this is a supervised approach, so you have to have a training data. And what we are going to do with this is, once you have these predicted genotypes, we know that they can be very noisy now. You can combine it with an unmasked data, such as the data from GED match, and you should be able to tell what the phenotype for this person is, because single-cell data usually comes with a phenotype. So this is currently what we are working on. So this is the quantification part. So we can use statistics and machine learning, information theory to understand what are the privacy leakages given a data type. But then what do we do with this? So we need to come up with solutions. How do we protect the private information? The first thing I did back in my postdoc, after we showed that there is privacy leakage in RNA-seq data, particularly functional genomics data such as attack-seq, chromatin histone modification, and such, we wanted to come up with a method that is easy to use for biologists. So what we wanted to do, so currently what happens is you have this functional genomics raw data. It's behind a firewall because of these privacy issues. You don't give any information to adversaries, but also it protects the data from scientists as well and create hurdles. So we wanted to create some kind of data sanitization such that the private component could be put behind a firewall, and then you give the sanitized data to the scientists, which they can compute any kind of information on it, and then it can be very accurate. So I did the sanitizer that divides the data, but it does it in a principled way where we can mathematically show that there is a balance between the privacy and the utility. And we even show that the upper bound of that utility loss when you have 100% privacy is very small. You can really, with more than 90% accuracy, you can calculate gene expression, for example, from RNA-seq data if you sanitize it. And then we also empirically show that it's pretty accurate. So here in blue is the difference between a sanitized and unsanitized data. And in pink is the difference between two biological replicas. So you cannot tell the sanitized data from biological replicas. So the noise added to the data is within the biological noise. So that's one way to do that. But the problem is with this is there is no theoretical guarantees of privacy, right? This is only you define your utility and you define your privacy. And it's only private for that definition. There could be some other ways to attack this data and figure out who the person is. So then we started thinking about some cryptographic approaches. And sorry about the weirdness here, but what I'm trying to say is that data protection is not used. In other fields, people have been doing that. So we have, data comes in three forms: data in transit, we protect it with encrypted connection; data at rest, we encrypt the data. The most problematic one is data in use, which is the data for genome. Like if you want to calculate something gene expression in cloud, there is a huge problem when you're doing this calculation. Somebody might be able to attack the data. And there are several ways to overcome this. The most known ones are the homomorphic encryption, secure multi-party computation, and Intel SGX, which is a trusted executive environment. I put federated learning here, but that's not necessarily a privacy tool, but it can help with privacy, which I will show later. The problem with all these rest of the three methods is that there's performance, applicability, and usability problems. And the genomic data is huge. The calculations we do are complicated and nonlinear. So it becomes a scalability problem, that can you actually apply these cryptographic primitives to the data. So first I'm going to, again, go back to my data science example, and I'm going to show you how we can do actually machine learning training in a sort of private way, and then I will tell how we can do the inference in a private way. So as I mentioned that in machine learning training, we have problems with the input, the private data, the training data, and now we have problems with the method, the model. So there was this problem of predicting broadcast status from gene expression data. And in this problem, we have three hospitals. They have their own patient populations with gene expression data. And the problem is that they cannot exchange data with each other because of the privacy issues. And then they can maybe do some model training together, but then they can't even share the model parameters because there's an issue with the model parameters. So here, what we did is we used federated learning to show that you can add noise to the model parameters. So federated learning works this way. You do one iteration of learning in each site. You send your model parameters, noise model parameters with differential privacy. You do federated averaging. You have a more general model. You send it back, and then you do another set of iteration of model training. And you do this until your final set of parameters converges to the same thing. So you're trying to create a generalizable model from the data across three sites without each site seeing their data. And it works really well. So here we tried different settings, different IID and non-IID settings with different number of samples and labels, and different privacy budgets. And here the lower the epsilon, which is the privacy budget, is the most private, and the most amount of noise you're adding. And in each setting, it worked really well. The reason it works really well is because gene expression data is very standardized. It's collected very similar way. Even though we are creating non-IID data here, it's still, the distributions are, like our genes act similar ways. You know, we have housekeeping genes. They are all expressed in the same tissues across populations. So there is a lot of data similarity. When you go to clinical data, it can be a nightmare, actually. So here, I'm showing that we had this prediction model where we are predicting this phenomena called delayed cerebral ischemia, which happens to the stroke patients, and it can be actually life-threatening. And it happens a few hours after someone gets stroke. At ICU, they monitor the people, and then if that happens, they have to put them in a certain kind of medication. And the idea is that can we actually make a model that predicts that as soon as the patient is basically admitted to a hospital, can you predict if this patient will have DCI or not? The problem is that this is a very small sample size because patient population, this is an ICU neurology patients, and plus they are the subset of the ICU patients who gets DCI. And because of that, we had very small amount of data, but we had full access to data from Columbia Medical Center, UT Health, and a hospital in Aachen. So we did federated learning, and now we did separate learning, and then we also combine all the data and do learning too. And we are comparing all three. The goal is that federated learning is very similar to the joint learning and better than the separate learning. But we found that in UTH and CUMC in Colombia, federated learning was great. But if you use the same model, federated model, in Aachen, it really worked terribly, basically. I mean, you can see even the error bars are horrible. And the reason for that is, even though we use the same features and same outcomes, Aachen was collecting the data in a different way, and the definition of DCI was very different for them too. So in clinical setting, we run into these problems. Then this opened up this whole new idea of, should we do actually federated learning, or it should be just single-site learning? And what do you do then if you have small sample size? So then we started talking about when to federate. When do you actually do that? So we were looking at, you know, what happens to AUC compared to single-site training if you have shared data sets. So we split the data into two, and then further split each data set into two, and then we have different sharedness proportion. And then as you can see, if you increase the data shared, you have a larger gain in AUC. But then how do you calculate if patient's population in one hospital is similar to the other hospital if you're not allowed to share the data, right? So then we thought maybe we can use the optimal transport cost, which brings one distribution to the another, as a metric to decide if we should do basically federated learning or not. Which was great that my student came up with this idea, because I know a person who is an expert in optimal transport, who is RITAMBHARA. So then an optimal transport is nice because there is a version of it where you can do an iterative optimal transport, which is perfect for federated learning. So what we do is we do optimal transport in a federated setting. Nobody share any data, but we share some kind of latent representation of data with some differential privacy. And then we developed a metric, optimal transport cost, that scales between 0 to 1, and it's universal across all the tasks. And according to that metric, you tell if the data set are similar, that if you should be doing federated learning or not. And we actually mathematically show that if the two data sets are completely random, you get 0.5 between 0 to 1. If the data sets are completely the same, you get 0 score. And if they are completely on the opposite side, you get 1. So by using this score, you first do federated optimal transport, and then you get this score. And then by using this score, you can decide which sites you should include in your federated learning, and for which site you should do more personalized learning. And we are now thinking of using the same idea for transfer learning, when to transfer and when not to transfer, when you transfer from one domain to another. And then this was the part how we are dealing with private information in training. And then now I want to talk about a little bit inference. So inference is a little bit easier in a way that you know exactly how many operations, mathematical operations you have to do, and you know the depth of your circuit. So you could do some more technical cryptographic tools for that. So we use, in my lab, mostly homomorphic encryption. Homomorphic encryption is the way that you perform computations while the data is still encrypted, so that if you have a cloud server, you can put a model there, and then your input can be encrypted, and then your output that you get is encrypted. And here, I love this example. It's Homer, because somebody apparently called homomorphic encryption they didn't know, and they called homomorphic encryption. And Kristen Lauter, who is now at Facebook AI, who did a lot of work in homomorphic encryption, she said, okay, I'm going to describe it using the Homer. So basically here, the cloud is Homer, who is performing on the data, and the jewelry is the data. And the data kept in a box. Homer puts his hands in, computes on the data, but he cannot take the data away. So that's how it kind of works. But there are a lot of hurdles. You can only do multiplications and summations. You have to create a data oblivious computing, so no ifs, no whiles. The flow cannot depend on the data. And if the depth of your circuit, meaning if the number of operations you have to do is really large, but at every operation you add noise, and when you decrypt it, you cannot anymore decrypt it because of the noise if it is above a certain threshold. And there's this technique called that Craig Gantry came up with, which is bootstrapping. But it's highly costly. It will never work with genomics data. So then what we need to do is we need to express all these bioinformatics tools with summations and multiplications that is already going to take away from accuracy. Plus, you cannot have a very deep circuit, meaning that no deep learning. You cannot do inference on like 20 layers of, even 20 is not a deep learning, but like you cannot do that. So I'm going to give you two applications examples. One is genotype imputation. Some of you might know that the SNPs, the genotypes we have in the genome are all correlated to each other. We have these haplotype blocks. All the SNPs are correlated. So if you know some of them, you can impute the other ones by using this statistical relationship. So then there was this task that if we have a genotype imputation model sitting in the cloud and we want to impute genotypes for a patient's genome, how do we do that with homomorphic encryption? So here, what we came up with is a single-layer neural network, because that's as much as you can do with homomorphic encryption. And in our model, the best performing model was a nonlinear activation, which had exponentiation on it. We approximated with Taylor series expansion, gave up on a little bit on the accuracy, and then encrypted the input, and then was able to get encrypted output. Surprisingly, this model works. Why am I keep doing it? This model works really well. So this is our model, AUCs, and these two are the state-of-the-art plain text imputation models. So we can have really good accuracy while protecting everything. So even though it's just a single-layer neural network, it works really well for this particular task. And then the another task we did was genotype-phenotype relationships. So if you, presumably you have a bunch of genotypes from patients, and now you have phenotypes. Phenotypes could be just 0 and 1, there is disease or not, or it could be some kind of continuous value that tells degree of some kind of disease severity. Can you create a model that is encrypted, that takes encrypted input and gives encrypted output? And in this case, we have a modeler that encrypts the data, puts it on the cloud, and then the client has a private data that wants to for a doctor has patient's genome and wants to know what is the probability of this patient having this disease. So in this case, we use 20,000 genotypes for 3,000 patients, and we had 5 different phenotypes. First 3 of them were continuous. The last 2 phenotypes were binary. And how do we create this data? This is coming from IDASH challenge, which is a genome privacy challenge that we attend every year. And how they created this data is that they use some complicated functions to come up with phenotypes. They used some simulated input and output and gave it to us. So in this case, we found that, well, we are working with very simple models here because of the limitations. But we created 2 different models. One, it was lasso, and the other one was logistic regression. But the nice thing is that in these two models is all you're doing is some kind of matrix multiplications, right? You're multiplying weight. This is an inference problem. You already have the weights. You're multiplying with the input. In logistic, you don't even need to think about bias, because when you calculate the AUC, you look at it's order preserving anyway, so then you can actually save some time by omitting the bias. And in homomorphic encryption works in polynomial structure. So everything is expressed as the coefficients or the degree of the polynomials. And doing multiplications, inner products with polynomial it's actually kind of easy. So you have one polynomial, another, you just basically sum all the coefficients, and then you get what you want to get as W and X. And how it works is you have a bunch of synapse and model parameters where you express them in the coefficients of the polynomials, and then you encrypt that, create a ciphertext, and then you do multiplications between two ciphertexts. One is the X, one is the W, and then you get the output. Output is encrypted too, and then you give it to your client, and local servers. They can decrypt it and then find out the results. So this is extremely fast and surprisingly accurate for this task. But it's actually, this is actually, you might think that this is not super fast, or it's very simple. Of course it's fast, but if you look at like 2 years ago with logistic regression with homomorphic encryption, we had to wait, I don't know, perhaps hours for 3,000 samples. So now we have this scalable, accurate machine learning in private domain. Yes?
Very quick clarification. This is the time to do the inference or the time to train the model?
Oh, inference. We can't, yeah, we can't do training.
Okay.
Sorry. Yeah. But this is promising because this opens the doors to more complicated, algorithms, and if there are any things that are done in a simple space, now we can actually use cryptographic tools for them. So with this, I'm going to end my talk by thanking my lab. The top 5 individuals, my 2 postdocs, 2 PhD students, and an undergrad student were the ones who did federated learning, machine learning for privacy, and homomorphic encryption. And the rest of my lab has been working on some other stuff. But I'm very grateful that in such a short time, like 8 months, I was able to they took a chance on me, so I really want to acknowledge them. And of course, RITAMBHARA and her students, it's been wonderful to collaborate with, having a blast. I couldn't find your pictures, so I put the lab pictures. And I have some funding from Data Science Institute, Columbia, and NIH, and also I was hired as part of Precision Medicine at Columbia and New York Genome Center. So, and thank you for all being here. Appreciate it.
Great. So I guess we can now move on to, well, thank you for a great talk, Gamze. We can now move on to the discussion section of the series. So,
Yeah.
I don't have a mic, but can people hear me?
Can you turn this off?
Wait, I just moved my chair this way. It's fine. I'll just move it.
I am still right there, so.
Let's try and shut this off then.
Oh, okay, great.
I did the wrong.
What is the line of facing? Yeah. Is that one?
It's not shining in the face.
No, it's not.
No. Do you want to sit there? Just.
So, let me start off with the first question, and then the audience can join in, and we would really appreciate questions from the students and the postdocs in the crowd. So because this is Data Matters, I have historically seen data science as doing inference, machine learning modeling, extracting patterns from the data, and now you are saying, well, privacy is a very important consideration for data science as the field. So where and how do you think privacy fits into the data science domain and framework?
Yeah, well, if you remember my slide, it's actually everywhere, right? The training data can be private. The output, the labels, and the model itself can be private, especially if you're using complicated models like deep learning. There's a lot of privacy issues. There's a lot of privacy issues in NLP, in medicine, where you go to clinical notes and you can accidentally extract some really private information, even if you de-identify the data. So I think that it always amazed me, data science institutes, same in my university or when I was at Yale, that was the same. It's heavy emphasis on machine learning, not as much privacy. Now I think we are catching up. So now, I think there was this boom of algorithmic bias, which is also very important, right? And then I think the next step for data science is to focus on privacy a little bit. So that's how I see it. And I think those are auxiliary to data science, but very, I think, important components, especially now we are at CCMB, especially if we are going to use health data or genomics, any kind of biological data. And there's also another issue that there is the changing landscape in legal structure, right, in different continents. So we will have to think about privacy, not just because it is important, but we will be forced, otherwise we won't have access to data.
I have a question, but now that we moved on to more general discussion.
I think it would be nice if you introduce yourself, your name, and what your department is.
My name is Pinar. I'm a fifth-year PhD student with RITAMBHARA in CCMB, doing computational biology. I think we moved on to more general discussion stuff, but I actually had a specific question about fancy.
Yes.
You had a plot that showed really good accuracy in determining how much
how many SNPs you can observe.
rare variants you would catch, right? But does that depend on the genetic diversity of the population in the data set?
Yeah, that's a very good question. Everything is trained and tested on European populations because I was using ATAC-seq data and ChIP-seq data, and unfortunately we don't have data from other populations. So yes, it will 100% depend on the structure because of how reference genome is determined. You typically see more SNPs in genomes from Africa, for example. So then in their functional genomics data, you will see more SNPs. But if you train your data, if you train your model like fancy with European data, then you're not going to capture all the variation. Yes.
My name is Emilia, and I am in ecology, evolution, and organismal biology. And in genomics, often people are, I think, they don't see why we should care about privacy with genomes. Why do you think we should care about privacy?
So there are two ways that I think. One, there's ethical responsibilities because the rules are changing, right? Like right now in US, we have GINA Act, which means that nobody can discriminate you based on your genetic information if they are doing employment or giving you health insurance. But they can still discriminate you if they are giving you life insurance, for example. But this can be changed. You can get a new administration anytime in US, by force or by formal election. And they might just say that, no, we're going to discriminate people based on their genetic. I mean, they're not going to directly say they are going to discriminate, but these rules might change anytime. And we are having more and more databases, so it's becoming easier and easier to figure out who is who and who has what kind of disease. So that's the ethical responsibility. The other is, as a researcher, that I have other responsibility in privacy space is that I want to increase statistical power when we do inferences, right? Statistical analysis. The only way to do that is to protect privacy because otherwise the institutional rules or even rules, between Europe and US, we're not going to be able to combine data sets. So that's why it's important too. So now there is another argument that I am in medical school now and I see how it is that the data seems to be a commodity in hospitals. So they often use privacy as an excuse not to share data. But then if you provide them saying that, hey, here is a mathematically guaranteed way of doing this, then you will remove that excuse, and they will either have to admit that they want to sell the data, or they will no longer have that excuse. So there are two ways to look at it. So that's how I look at it.
I'm Suresh. I'm sadly one of the computer scientists who does remember the Netflix price. I'm old enough. One of the things that has come up in other areas, so I'm curious how you see it in your domain, is that a focus on privacy and the kinds of modifications to the data, like say using differential privacy, that you might have to do to the data to preserve privacy, actually make it harder to do a proper analysis of bias, mainly because small groups like indigenous populations are already in very small amounts in the data set, and to preserve privacy, you have to add a lot of noise, and that lot of noise actually swamps the signal out, and so it makes inferences for those groups even harder and therefore more biased. Have you seen this phenomenon in your domain, and you see people trying to address this?
Yeah, 100%. If you do federated learning, for example, the one dominant group will dominate your model, and then your model will be extremely biased. So if you're doing federated learning across, let's say even in New York City, across Columbia hospitals and NYU, they sit in different parts of Manhattan. They have very different demographics, and whichever the demographics is the dominant, then you will dominate your general model. So that's why I think that people are, when to federate, we want to answer that question too. So coming up with a metric where you can tell that, oh, there is the group, like putting some kind of safeguard. There is this other data set that's coming from this different distribution, and perhaps for that distribution, for that data set, we should do some more personalized modeling. So that's one way. Another way is that with the hope that the cryptographic techniques will catch up with the growing amount of data, where you don't have to worry about that because you're not noising the data. You're just basically doing everything in the encrypted domain. So that's another way, but that we are long ways away from that. So it's a very difficult task.
Hello, I'm Julia. I'm from philosophy. I'm interested in tech ethics. So you said at the beginning that one reason why people who work with genomic data should care about privacy is because it's essential to maintaining trust. So to making sure that people are actually willing to let you use that data, to consent to using that data. And I was wondering, to which extent do the methods you are using, to which extent on how are they building this trust? Because these are all very complicated techniques that probably the average person in the hospital who, or in whichever setting you ask them to donate their data, will probably not appreciate. So how does this help build trust? Yeah,
so this is not short-term trust. So it's building long-term trust. So if we do the things the way that we are doing, we will see leakages, right? There will be some catastrophic leakage sometime, and then it will be said on news like, oh, UK Biobank, now we have access all the data. And this happened in the past. So by building trusted algorithms, basically the cryptographically secured algorithms, then you're avoiding that long-term catastrophic leakages, that you're building long-term trust, basically. Then people are saying that, okay, this is not, it didn't happen in the past 10 years because they were telling me that they are using secure algorithms, and now I believe that this is happening. So it's more like a long-term goal than the short-term. Otherwise, the informed consent is complicated as it is. Adding it that, oh, I'm going to protect the data with homomorphic encryption, nobody's going to, that's not going to work out.
I'm Margaret from the neuroscience department. So if you're, in a practical way, if you're running a clinical study and you are dealing with different data sets coming from different centers, how can you implement this, and how easily is to train people to implement this method? And so I have this problem. You would suggest me to look into this right now, because this is not required now within the management system for privacy, right? From organization, including Brown, right?
Yeah, no, that's a very good question, and it's a big problem because this is very domain specific. It's even within the domain. So there are security and privacy people in CS, and then there are privacy people like me who work with the biological data. Even us speak different languages. So that I cannot imagine going to a clinical center and saying, hey, why don't you do this in a federated setting? they will look at me and they will just say, like, okay, our life is as hard as it is, so you're going to add one more layer. Yeah, so I think that we are making baby steps. So it's really proof of concept, theoretical, and with the hope that maybe 10 years from now, we will start having, nice user interfaces and very good front ends where people can do things with one click. You can provide these, and in the back end, everything runs in encrypted domain or in federation. So we started doing that in my lab, where I actually contracted a software engineer to build a front end with some system that we are building for combining clinical data with the genetic data. The problem is that it's a lot of thankless work. So I spent all that money to get maybe one paper out of it, use a part of my startup or some federal grant, and nobody actually cares if it's usable or how easy it is for people to adopt. So that's a general problem, I think, in the field, where people are developing things to publish, not for use. Yeah, so and same problem here too. And here a bit more exacerbated, right? Because I'm telling the people, you should use these things. You have privacy problems. If I don't provide them to an easy way to use them, then I actually don't have the right to say such. So.
Privacy protection a significant hindrance to precision medicine? How much more farther along would precision medicine be today if there weren't privacy protection?
Also, is privacy a problem in precision medicine? While they say that yes, because of privacy, there is not much, advances in precision medicine, but in reality is because, as I mentioned, that the data is commodity in hospitals. So they actually hide behind privacy when they don't want to harmonize. So in precision medicine, what we want to do is we want to find the right cure for the right person at the right time. But then you have to have some other examples in your data sets that are similar to that right person so that you can make connections, right? If you have a rare disease. But if you don't have access to data in another hospital, then you won't be able to do that. And that access could be genuinely because of privacy issues, but it could be just because the hospitals don't want to share their data because of the issues related to monetary part of the industry.
I have a question. So I just want to thank you. First of all, I'm a faculty member here in DSI, and I have no biology background, so your talk was very accessible. So I appreciate that.
Thank you.
And so one of the questions I have is, you talked about your work where you're kind of showing some vulnerabilities to these data sets that you can kind of trace who the people are. And I guess I'm curious, have there been any real world examples of that happening that were widely publicized, or is this something that you're sort of worried about in the future, or is this something that maybe I've missed it and there was some big news story where something like this happened, but I'm just curious.
So I think, Netflix Prize is one of them, which is not biological data, but I think that was one of the real world examples. There was another one where there was a science paper about figuring out the last names for individuals. So there's this data collection we call 1,000 genomes, where we have 2,500 genomes from different populations, and some of them are European population, and these people are particularly located in Utah. We know that. And this data available everywhere. And this individual, Yanni Verlick, who used to be actually at Columbia, he basically developed this statistical method where he said that, Y chromosome is in men, and traditionally last name propagates from men in the family. So I'm going to use these people's Y chromosomes, the mutations on the Y chromosome, go to these databases where I have these people's names and addresses, and find out who they are. And he actually figured out a bunch of people from the 1,000 Genomes project to their zip code in Utah. Of course, he's not showing it in the paper, but so that paper made a big splash, for example, that you can find. And then he also figured out that if you use all the data, so he used to work for myheritage.com, which is an ancestry company. He used all the data there, and he said that if this data is out, 60% of European American population is identifiable because it's all there. So these are, so I think the last name, surname inference was kind of a real world application of that. There's also, China uses the 1,000 genomes data and some other data to kind of create profiles for different ethnicities in the government using that. And they are not hiding it, right? It's public information. So data is public, it's fully consented, but there are some cases where it can be used for, nobody would have thought that the 1,000 genomes data would be used by Chinese government for racial profiling when they were sharing that data. So it was very innocent, right? We wanted, it was a big population genetics project. It was great that we had all these access to this data, but 10 years after, you see that there's misuse.
So some of these examples you mentioned are very concerning because, people have been sending their DNA to Ancestry and 23andMe, and now you are coming here and telling us if people have access to this data, they can find things about you that are very private, you won't want to share. So given all these concerns, what do you think as far as research in the domain is looking into the future? Are there things being done to reduce these concerns about what happened? I have submitted my DNA, there's nothing I can do anymore. How do I sleep at night?
Yeah, so I believe there are checks and safeguards in these companies that, they are not stupid, right? They are making a lot of money from the data. So I'm sure that they have safe warehouses for the data and all that stuff. But when they do computation on this data, everything is in plain text and it is vulnerable. I think in the future, first, there needs to be more oversight, right? It's not clear if this data comes out, what happens. Yes, there were some data leakages in other domains that happened. So what happened? Everyone forgets about it after 5 days. It becomes a big news. So that's not very clear. And because of that unclear nature of it, I think they are not so concerned. Maybe regulations. Not a big fan of regulations and rules because they change. So I think we need to mathematically protect the data, basically. So that's my job, right? But again, the technology is not there yet, but I think we are making little by little steps. Like, for example, when I did the data sanitization, the ENCODE project that we were working, they wanted to share this data collection called Roadmap, but they couldn't because the people didn't consent, and most of the people were already deceased, so they couldn't share this data. So they took my algorithm, did data sanitization, and now all that data is available. So now we know that there's some kind of protection, but at the same time now researchers are accessing the data and doing a lot of great things with it. So as we develop these tools, I think that in the future, but people are resistant, right? They want to do the things the way that they want to do, so there's also that.
So it's a long way to go.
Yeah.
So coming to regulations, actually, you have interacted and collaborated with researchers in the EU. They have a completely different ideology on privacy, and then you have worked here in the US. What differences do you see between the way privacy in research is approached across these two blocks?
Yeah, it's completely different. I don't think it was this different before, but with GDPR, I don't know if people are familiar, but it's a new privacy rule in Europe that makes life very difficult for researchers. You can't share data. Even anonymized data cannot be shared unless it's encrypted. But even what do you do with encrypted data? If you don't know what is in the data, what can you do with it? In US, it's completely different. Anonymized data can be shared. We have checks and safeguards like IRBs and stuff, but once you have access to data, nobody cares what you do with the data. Nobody asks you anything. You just have the data. So Europe is a complete different way of doing things, and if you want to collaborate, we kind of have to, do the things that they do, because otherwise, like Human Cell Atlas, a lot of people use single cell data. They have a huge problem because half of their donors are coming from Europe, and they don't know how to share the raw data, and they don't even know if the summary data has problems. But then US started doing things differently, like California, because of Silicon Valley, they completely said, okay, I'm going to do whatever the Europe does, because otherwise I can't collaborate. So they have their own set of rules. In New York State, we have, even though in the country, if you give your genomic data in an anonymized form and use consent for it, then that data can go to public domain. In New York State, you can't do that. It has to be still people has to apply for access. And this heterogeneity in US makes it very difficult to collaborate, to share, and forget about Europe. I think Japan has very strict rules as well. But then there are some countries where you are born and immediately genetically profiled and put in a database. So, yeah. I don't know if this answers your question.
No, it does. So it basically comes back to the point of like having trust in the systems where, it's not only about sharing data at a local level, but if we have to do any sort of global collaborations, that trusted system needs to get in place so that people can share data and do research on a...
So they started launching these cloud enclaves, right? Like UK Biobank has this system called RAP, I believe, where you can't take the data out. You have to do all the calculations within that enclave. It's not completely secure, but it's one layer of security, which is kind of nice.
All right. If there are no other questions from the audience, I can wrap it up with one final very important question. So actually, there are like two sub-questions. So for students and postdocs who want to work in this research topic with, which has broad implications and interests, what kind of training do they need to do? And do you have, given that your own career trajectory has led you over here, what kind of advice would you have to the trainees as they navigate their career?
I don't know if I'm the right person to ask this because I had no formal training in privacy. I did my undergrad in chemical engineering and then became a bioinformatics person. And then when I was doing bioinformatics, it was mostly statistics. And I realized I was doing regulatory genomics, but I didn't understand how genetics works. So I wanted to learn more genetics. But then I realized like everything was already taken, that was like 10 years after human genome was sequenced. So I'm trying to find something that I can do that is different than others, but also will teach me a lot of genetics. And that's how I came to privacy. But I also, of course, had personal interest in privacy. So if you look at this example, then what I would say that you don't need to have formal training if you want to work on this field. I think, like I'm hiring, for example. So and if you look at my lab, people who work on privacy, none of them have any background in privacy. But they are learning with me, and I'm learning from them. They have expertise in other stuff. So PhD, postdoc, it's a little different, so I'm not a person who learns in the classroom anyway. So I'm a person who does the stuff and learns while doing it. And if you're a person like that, it doesn't matter which background. It's like philosophy, you can do privacy. So I think for this particular type of projects, if you know how to code, I think you'll be fine. So
they all need to just do coding.
Yes. Well, I mean, because that's the hardest, I would say, activation barrier. Like that is a difficult thing to teach in a lab. So you have to have that formal training, how to code. But then after that, you just learn by doing and reading. And it's a new emerging, field. If I look for people who are experts in genome privacy, there are probably 3-4 labs in the world. So I can't find anyone. So I have to go with people. I was one of those people who didn't have background. So it's an emerging field, and that's why I think it doesn't matter. Mostly computer science, computational biology, that kind of training, I think will be enough for this.
All right, so on that positive note, thank you everybody for joining in. Thank you for giving such a great talk and
thank you so
answering all
much.
the questions. And a round of applause for us.
Thank you so much.